Get slides up there. Perfect. Amazing. So thank you for the introduction. I'm Jackson Pullman. I'm a researcher with the Human Nature Lab at Yale University. Broadly, for some background, our lab's work lies at the intersection of the biological, the social, and the computational sciences. But the main questions that many of our projects engage in are how and why do we humans assemble ourselves into social networks, and what does this mean for our lives? We've been working to understand the genetic and evolutionary determinants of social interactions, as well as their biological implications for over 15 years. And over the last 3 years, we've been making heavy investments in work with the microbiome, exploring how our social interactions have the potential to shape our microbiota. So there has been some past work on how the human microbiome can be impacted by our social relationships. For instance, it's well established that maternal transmission to offspring is a primary seed of the infant microbiome, and that individuals living within the same household tend to share more microbial elements than individuals across different households. However, the influence of face-to-face social interactions beyond household contacts and closely related kin on the composition of the human microbiome is still incompletely understood, yet surely relevant. So we have a number of overarching questions for our project, all generally related to the idea of whether a microbial species or strains are distributed randomly across the social network, or to what extent can a social network explain in any way the distribution of the human microbiome. And discerning how the microbiome might spread across social ties might allow us to see that diseases previously thought to be non-communicable, such as depression, obesity, or hypertension, might actually be communicable through the microbiome. So we've built a data set centered in the Western Highlands of Honduras to investigate these questions, which is part of a larger population-based cohort that we started following in 2015 for a different purpose. Our first round of results involved 9 villages and 1,098 participants, which will be expanding to 19 villages and 1,887 participants within the next month. And generally, our village sizes in our study range from 89 to 432 individuals. So we set up labs in the field to collect stool from microbiome samples, and we performed a strain level profiling with strain flag 4 to detect potential transmission events between pairs of individuals. And we can summarize the strain level similarity between any 2 individuals with the strain sharing rate metric, which is equal to the number of shared strains between 2 samples divided by the number of shared species. And then on the social network side, after a village level census of all these villages, we can discern social ties via name generator questions. So we ask all the village residents questions such as, with whom do you spend free time? Who are your closest friends? And after this whole process, we can identify 2,919 unique social network links, which span multiple familial and social categories. So kind of in sum, our data set combines face-to-face social networks, a set of individual and community level characteristics, and gut microbiome sequencing data, resulting actually in the largest ever cohort in terms of enrolled participants and sequencing depth in a population from a non-westernized setting. So within our villages, we observe microbiome strain sharing occurring across many relationship types. Here, the x-axis denotes the relationship bucket, and the y-axis is the strain sharing rate for pairs of that relationship. For context, the 2 box plots on the far right contain the strain sharing rates for pairs with no relationship, but who are living in the same village, and for pairs who are living in different villages, respectively. A number of features are apparent. First, spouses and relationships between individuals living in the same building, the 2 box plots on the left-hand side, have the highest levels of strain sharing. And also, as we've seen previously, mothers tend to have significantly more strains shared with their children than fathers, potentially as a result of infant transmission. And generally, we observe a kind of social distance-based gradient of strain sharing. Interestingly, we also observe a significantly elevated strain sharing rate between people who are neither kin nor who are living in the same house, but do have a social relationship, the 4th box on the right. And there's minimal amounts of strain sharing between individuals who live in the same village, but don't have any social relationship between them. For a subset of these relationships, we also collected data on how microbiome strain sharing may relate to how often the pair spends free time together or share meals together, represented by figures B and C respectively. For both of these categories, people who interact almost every day, the box on the far left-hand side represents share more than those who interact only once a week or a few times a month. And both of these results also hold when considering non-kin relationships who are living in different households, suggest that these types of contacts are potential transmission routes of microbiota. I think some of my slide, the images might have gotten compressed a little bit, so hopefully it's not too blurry. So we've seen that individuals are more similar to their friends than we would expect by chance, but we can also predict that because of this, they're more similar to their friends of friends. and such ideas would be consistent with microbial spreading in a population or potential chains of transmission. So we can calculate the strain sharing rates by the degree of separation between any two people, called the geodesic distance here, which is on the x-axis, with the observed rates represented by the decreasing blue line in this figure. To construct a permutation null distribution under the null hypothesis that an individual's microbiome composition is independent of their social network's microbiome composition, we can randomly swap the microbiome of every individual in the village and recalculate our metric of interest, which is represented by the red line in the figure. So we see that first-degree relationships have a much higher strain sharing rate than we would expect by chance, but this effect also extends to second-degree social connections before falling off at the social horizon of our third-degree social connections. There's no higher strain sharing rate than we would expect by chance. So these kind of patterns suggest that we can view microbiome transmission or microbiome sharing from the framework of disease ecology, much like how socially central people in a network may be more easily exposed to a cold or flu that's going through a network. We might also expect that individuals who are most socially central may be more exposed to circulating strains in the network, what we could call kind of the social microbiome of the villages. Conversely, socially isolated individuals may be microbially isolated as well. So we can measure how similar someone's microbiome is to everyone else by calculating their average sharing rate with everyone else in the village. Then we can measure an individual's social network centrality in multiple ways, some of which are degree centrality, their absolute number of connections, your betweenness centrality, or how likely you are to connect people in a network, and your eigenvector centrality, which is a function of your number of connections and the centrality of those connections. So after controlling for different socio-demographic variables that could be related to any of these, we see that all measures of your social network centrality are positively correlated with your centrality in the microbial network. Relationships shown for just degree centrality here. So for the more kind of socially embedded someone is in a village, the more microbially embedded they are as well. These ideas kind of represented graphically in the figures on the right-hand side, when nodes are sized according to their degree centrality. And the nodes colored red are more microbially similar to everyone in the village and also tend to be bigger in the center of the social network, while those that are colored yellow tend to share less and are smaller and occupy the periphery. So human social networks generally have significant clustering. You can think of like kind of groups of friends or your family, and each node tends to lie in a different region of the network. We may expect that this will also be important from microbial development. So we can kind of consider the analogy of studying the bacteria if you went outside and sampled dirt from different patches in the ground. We'd expect to find differentially abundant bacteria in each patch, and the species present there could work like a colony of helping each other, maybe potentially passing mobile genetic elements. So the species in each patch would also depend on the water, the sunlight in the patch, the debris. So similar to this dirt example, we can imagine that the collection of species might vary from patch to patch in the social network. And if so, we would find significant strain clustering within the network. And this clustering might arise in the spread of the microbiome across social ties and also might have deep evolutionary pressure may relevant to it. So in figure A, in the top left, we can see at the smallest scale, individuals with a higher clustering coefficient, that is, individuals whose social connections are also social connections with each other, tends to encourage a higher average strain sharing rate amongst those people. So having connections who are also connected may encourage microbiome circulation within the cluster, reinforcing kind of microbiome niche within the social network niche, whereas individuals with links to many different parts of the network do not experience this reinforcement effect. To observe this phenomenon at kind of a whole village level scale, we can identify both social and microbiome clusters with a technique called Louvain clustering on the graphs, which can work on social network data as well as biological data. So an example of microbiome clustering based on the strain sharing rate between individuals in the village is shown in figure B. An example of social network clustering just based on the social ties is shown in figure C. We can then paint this microbiome cluster membership onto the social network clustering and visualize the correspondence between social communities and microbiome strain communities. And across all these villages, social clusters visually overlap with microbiome clusters, which we can see in figure D, where the nodes are actually colored according to the microbiome cluster, but these can almost nearly perfectly visually overlap with the social network clusters. And there are a few methods we can use to show that these are statistically significant overlaps between the clusterings, suggesting that generally your social network community is also your microbial network community with potential deep importance for human health. We have a lot of acknowledgements, people to thank. This study has been going on for a few years. So of course, everyone in Honduras, all the study participants, local organizations that we worked with extensively, including the Honduras Ministry of Health and the Inter-American Developmental Bank, who we worked with to ensure that these studies were all done safely and equitably with high regard for privacy. Some of the names are listed there, as well as all the supporters, both within Yale and our partner lab at Cornell, the Brittle Lab, as well as our funders, some of which are listed at the bottom there. So thank you very much.